Well, good afternoon, everyone. Welcome to the 2021 annual meeting of the Western Regional Panel on Aquatic Nuisance Species. I'm Dennis Sabaglo. I'm the Aquatic Resources Program Manager for the Tahoe Regional Planning Agency, and humbly serve as the chair. So thank you for being here and welcome. And 2021, what have you done for me lately? Not a place where I think we all thought we would be. I think we were all hoping to see each other in person this year for our meeting, but given the circumstances, conducting a meeting virtually is really the most prudent. But that being said, a lot of work goes into developing our agenda, and the planning the meeting even virtually is a lot of work. So I want to please join me in thanking our planning team. Tammy Davis from Alaska Department of Fish and Game, Nate Owens from Utah Division of Wildlife, as well as Bruce Johnson, also with Utah Division of Wildlife, Tom Booz from the Tahoe Regional Planning Agency, Tom Wolf from Montana Fish, Wildlife and Parks, Heidi McMaster from the Bureau of Reclamation, and Chris Shiani from the California State Lands Commission, Elizabeth Brown, I think some of you might know who she is, from NASMA, and Leah Elwell from Invasive Species Action Network. So thank you all for making this happen, and we look forward to seeing you next year, hopefully in person. And then I would also like to thank our executive committee members. A lot of hard work goes into making sure our panel continues to achieve the goals that we've set out. So Tom Wolf, who serves as vice chair for Montana Fish, Wildlife and Parks, Glenn Dolphin from the Oregon State Marine Board, Martha Volkoff from California Department of Fish and Wildlife, Blaine Parker from Columbia River Intertribal Fisheries Commission, Barak Shamai from U.S. Fish and Wildlife Service, John Wolschlager from the National Park Service, Allison Zach from the University of Nebraska, and our infamous Quagga czar, Stephen Phillips from the Pacific States Marine Fisheries Commission. So thank you for your dedication and your hard work to ensuring the continued good work of our panel. And I also want to thank all of you for being here. Not only for being here, but continuing what you do during really kind of continuingly challenging times. Not only do we face threats from what climate change and catastrophic wildfire can do to the waters we strive to protect, but also the continued stresses from increased demand on those resources, be it recreational or otherwise. And then on top of all that, having to navigate how to continue our important work during a global pandemic. I'm truly awed by all the great work all of you do, so thank you. And we should all be proud that we continue to accomplish at the WRP. We provide leadership at the national level in the fight against AIS. We lead the effort on Disseminated mussels and coastal issues, spur innovation and control, minimizing spread from critical wildfire fighting methods, investing in research and tackling complex issues like eDNA, and ensuring education is at the forefront of everything we do. It's really our most powerful tool we have in our efforts. And then there's also a lot of work from an administrative and operational standpoint to ensure that based from those committees that help WRP operate efficiently and effectively. And also thank you to the Invasive Species Action Network for coordinating everything and keeping us on track and continuing to improve our operations. So thank you all. Very strange times, but I'm hopeful that we'll all be seeing you in the great state of Alaska next year. So fingers crossed that things continue to return to some level of normalcy. So before I turn it over to Leah to talk about how the meeting will go, and then we'll get started right into our agenda. But I want to personally thank all of you that reached out to us here in Tahoe. I'm sure a lot of you are aware of the Caldor fire that came in through here. Just really grateful for the thoughtfulness and the generosity all of you offered during these times. So I'm really grateful for the WRP family. So thank you all, and here's to a great meeting. Leah?
Awesome. Thank you, Dennis. It's good to be part of the WRP family. Well, welcome everybody. Good afternoon, wherever you may be. Hopefully you can see the agenda here displayed of what we have in store for you today. I just want to go over some critical items related to the meeting today. So, and this is directed only at our presenters, so I will be keeping track of time, and if I need to, I will interrupt you, but I know you're going to do lovely, smooth presentations, and we'll have plenty of time for questions. As for questions with our audience, we would love to field any questions that you might have for each of our speakers today. To help me kind of facilitate that question and answer period, if you could please put the name of the person presenting in front of your question, it just helps me track and make sure I'm directing it to the right person. These webinars for every single day this week will be recorded, and likely towards the end of next week, they should be posted on the WRP YouTube channel. So if you haven't already liked that or followed that YouTube channel, you can go there and find actually the broadcast from the previous last face-to-face meeting that we had there too. So, and then just a couple of other items for the rest of this week as well. We are interacting on the WRP Facebook page. If you haven't already liked that, we're just kind of hanging out and sharing information there. And then this morning we had our coffee hour, which obviously is come and gone, but you can still join some social interactions with us. I know not everybody that's attending can put on their webcam or, you know, unmute themselves. So we have the opportunity of Thursday at 5 p.m. Mountain Time. You can join for happy hour and we can chat and tell jokes and play bingo like we have scheduled. So all those links are found on the website. So the westernregionalpanel.org, and just go to the virtual meeting page. And you can find the links to join that happy hour and also info on the Facebook. So also, to anybody that's attending this week, I will be sending out a meeting survey to get your impressions about what you experienced and to get some thoughts about next year as well. So look for that survey from me. And then I will put it in the little chat area here now. But if you would like to be on the WRP listserv, so if you're attending and you want to know about news from the WRP all year long and get the monthly e-newsletter, you can get on that, no problem. So all you have to do is send me a quick email saying that you want to do that. So I'll just pop my email in there, and also a link to the website for those social links. So with that, I would like to hand over the voice box to Mr. Tom Wolf, who will be moderating this session for us with Montana Fish, Wildlife, and Parks. So welcome, Tom.
Thank you, Leah, and thank you, Dennis, for that great introduction. My name is Tom Wolf. I am the Vice Chair of Western Regional Panel and also AIS Bureau Chief for Montana Fish, Wildlife, and Parks. We are kicking off the session this afternoon, AIS Innovations and Technology, with our first speaker is Nate Owens. He is the Aquatic Invasive Species Coordinator for the state of Utah, one of my personal heroes. This guy has done incredible things in Utah to really step up the program down there to address the threat of things like zebra and quagga mussels. The issue at Lake Powell has been significant, and he's worked really closely with Park Service and a lot of other partners down there to try to contain that threat. If you weren't down there, I think we'd be seeing mussels in a lot more locations nowadays. So he's doing great work down there. We're talking about technology and innovations here, and he's also led a lot of efforts to address advancing technology to improve containment and decontamination of vessels. So Nate, take it away. Great work. Thank you, sir.
Thank you, Tom. Does that look good?
Perfect.
Okay, awesome. So again, thanks, Tom. Hello, everyone. I wish we could do this in person, but I miss seeing you all, but I guess this is what we have for now. As Tom mentioned, my name is Nate Owens. I'm the Aquatic Invasive Species Coordinator and the Boating Access Coordinator for the Utah Division of Wildlife Resources. And I will be presenting on Taking the Dip, Rethinking Watercraft Decontamination. And before I get started, I would like to point out that I first mentioned this idea to the whiskey group at my first WRP meeting at Lake Tahoe way back when. I'm sure at that point, most of you thought, who the heck is this and what has he been drinking? Well, here we are. So most of you in the decon business, you'll probably have a few emotions come to mind when I show you a few of these things. So when I show you this, or perhaps this, or this, definitely this on a not so good day, or this, and let's not forget this. Or if you are working on a launch ramp and you see an owner of one of these boats come up ready to launch, you think to yourself, are you sure you want to launch here? Don't you want to launch at that reservoir down the highway so I don't have to deal with your boat? Don't worry, we've all been there. So most of us are kind of thinking the same thing, unless you have interdiction specialists and decon all-stars on your team. Luckily, we do here in Utah. I know a lot of you do as well. But let's not forget that most of the people doing the decons are seasonal technicians, and that is a giant pain in the butt. So let's just kick it off with going through the decontamination process so everyone's familiar with that. Most of us obviously know it inside and out, but boats recently used in zebra, quagga, mussel waters are often professionally decontaminated through the application of hot water. 140-degree Fahrenheit water to all external areas of the watercraft and the engine for an exposure time of 10 seconds. And 120-degree Fahrenheit water to all internal areas and other raw water systems on board for an exposure time of 130 seconds. So all ballast systems, engines, showers, any other raw water systems on board are flushed with this hot water to kill any mussel veligers that may be present in a lot of these systems that can never be fully drained. And then the external areas are sprayed with that high pressure in some cases, or low pressure in some of these other areas, with that 140-degree Fahrenheit water to kill anything that may be attached, whether those are juvenile or adult mussels. Decontamination can be very simple, but it can also be very complex. The time and training required to not only train people to do the decontamination, but then actually perform the decontamination itself can vary widely based on the type of boat. So on the left, you see a simple fishing boat. Pretty simple. You know, we can probably train someone on how to do a proper decontamination on this boat in a few hours. And then the decontamination itself probably can be done in 5 to 10 minutes fairly easily. You look at this boat on the right, obviously that's a much more complex boat, a bigger boat, a boat with several internal systems, ballast tanks, those types of things, the things that you see on some of these wakeboard boats that are becoming more popular. Obviously, this is going to require a lot more time, a bigger investment in training in order to really get people up to speed and comfortable with decontaminating one of these things. Decontamination training will probably include a full day at minimum on how to decontaminate this, but also probably watching someone else decontaminate this boat several times before someone else feels comfortable doing it. Let's not forget also that you have all these attachments you also need to worry about. The fake-a-lake to try and get the hot water up into some of these ballast tanks that we're all so concerned about. The muffs to get it into any outdrive that you need to get water up into. All these special attachments that, depending on the type of boat, the boat manufacturer, you might need this attachment or that attachment, or you might have to jerry rig this thing. There's all sorts of things that go on when you start talking about some of these more complex boats like wakeboard boats. As you can imagine, putting a technician on the ramp and having that and basically telling them, look, you're in charge of doing all this, most of the time they're going to have to figure it out as they go along. That's a really tough position to be in, especially when that person may only be 18 or 19 years old. Put them someplace down like at Lake Powell in 110-degree air temperatures. Yeah, you're probably not going to make a whole lot of friends. So most of you know that Lake Powell is kind of the center of what we primarily do in our program. It's the site of the largest invasive mussel containment program in the country. We have awesome partners down there, the National Park Service at Glen Canyon National Recreation Area, Arizona Game and Fish Department, and then we do a lot of our work with funding from the Bureau of Reclamation and the Fish and Wildlife Service. This graph over here on the left shows the statewide decontaminations by year done in Utah over the last 7 years. So as you can see, that the number of decontaminations is steadily increasing statewide every single year, and we don't see that slowing down anytime soon based on trends. Almost 12,000 decontaminations in 2020. Over 4,000 of those were just at Lake Powell. So you can imagine that the number of decontaminations being done by every one of our technicians just at Lake Powell alone. So we have a pretty big problem. First problem is, how can we decontaminate more boats at Lake Powell, given the fact that we expect to see greater and greater numbers of people going there with their boats? The second one, boats seem to be more complex every year. Every year we see some crazy new boat with some crazy new system that requires, 3 different inputs of water to decontaminate one system or something like that. How do we stay ahead of the game without continually finding ourselves struggling to figure out how to address some of these boats? Can we simplify the decontamination process in any way? We seem to be getting more complex, and that's not necessarily always the answer. How can we increase the effectiveness and our confidence in the decontamination process? So most of you are probably aware that when it comes to wakeboard boats, most of the time we don't have the water required to completely fill those ballast tanks full of hot water, nor, if we're working in a busy area, nor do we have the time to do that. A lot of our machines pump water in at 5 gallons per minute. Let's just be conservative. A 50-gallon ballast tank then will take 10 minutes to completely fill, and there's probably 3 or 4 of those on board. So we can't really do that when we have a line of 20 boats waiting to get in. And then finally, how can we reduce or potentially even eliminate a lot of the specialized training required to adequately perform a decontamination? Again, a lot of our programs find ourselves in this kind of battle of not only a struggle to hire people to do the job, but also to retain those people. It's a huge investment getting these people trained up to be able to do these types of decontaminations. The last thing we want is to have someone quit in the middle of the year and have that investment go out the window and then have to redo it all over again. Is there a better way to do this? So many of us at the Utah Division of Wildlife Resources, over the last 8+ years, we've been talking about different ways to decontaminate boats because we knew that this day was going to come. And the most popular idea kind of revolved around this concept of a giant bathtub for boats, where a boat could just be back down in there. Everything could be operated the way it's meant to be operated on a reservoir. All those systems could be decontaminated all at once, and we could remove all these specialized attachments and all these other variables. We had what we thought was a great idea, but there were also a few problems. One, we didn't have the funding to do it, which obviously that's a huge one. Secondly, we didn't have the technical know-how on how to make it happen. And then third, quite frankly, there wasn't the urgency, 5, 6, 7 years ago. At that point, we seemed to have Lake Powell mostly under control. Obviously, the last couple of years at Lake Powell, we've been facing challenge after challenge, and these have all been turning points. So we kind of knew just within the last year that we had to start evolving quickly. So in 2020, with generous funding from the Utah legislature and the US Bureau of Reclamation, with obviously a ton of help from the awesome team at the National Park Service at Glen Canyon National Recreation Area, we were able to partner with 2 awesome, really solid guys that are just problem solvers with Clean Wake, Garrett and Jonas. And you'll probably be hearing a lot more about these guys down the road, and I encourage a lot of you to reach out to them directly. But we began hashing out what this kind of system would look like. What are the details that would have to go into such a system to make this successful, not only for, you know, decontaminating boats, but also the people that are going to be tasked with operating and running this thing once it's on the ground. So some of the details, and I don't want to get too wrapped up into this, again, contact me offline, but we're talking about a propane generator to power a skid-mounted commercial-grade filtration system so that we make sure that any villagers discharged back into the tank water are filtered out before another decontamination is done. A propane heater to maintain that tank water temperature at 110 degrees Fahrenheit. All these different, you know, things that I never would have thought of. Anti-skid traction plates on the tank itself, heat retention paints, just redundancy in the system, shade balls that can reduce evaporation and minimize algal growth in the tank. All these things that kind of went into this, and Garrett and Jonas worked through each one of these and figured out a way to implement it. And since this had never been undertaken before, we kind of knew that we were just going to have to make adjustments on the fly and figure it out as we went along. But we also thought that was probably a lot of the fun and something that would be very rewarding. So when it came time to actually start thinking about putting this thing together, we wanted to do one external review and see what we were missing. And obviously, the one thing is we needed expertise from the manufacturing community, and namely the trailer manufacturers, the boat manufacturers, and the engine manufacturers. Was there something we were missing when it came to designing this thing that we really ought to look at, some sort of fatal flaw? So once and I'd like to thank Brian Goodwin for setting this up. The American Boat and Yacht Council allowed us to present this concept to their community, and it became very clear that one thing that they were concerned about that we had completely overlooked as the alternative need for a decontamination method for the bilge area. We had originally just kind of incorporated the bilge area into this, you know, submersion / flooding method through the dip tank. Obviously, the manufacturers didn't feel that was a wise move. With a lot of the electrical back there, they felt it was too great a risk. And so that was awesome input. We took that in stride, we made the adjustments, and we ultimately came out with a better product. So fabrication begins. Super exciting. When we saw that first, the tank was absolutely humongous. It was 42 feet long, 14 feet wide. At the deep end, it allowed for a water depth of 5 feet. It can hold approximately 9,000 gallons of water and can accommodate boats roughly up to 26 or 27 feet in length. So once the tank was fabricated and all the other associated systems like the heater, the generator, the filtration system, all those things were acquired and installed, we tested the system at the manufacturer site before transporting it down to Lake Powell. It was a pretty cool thing. We did a huge media event. Pretty cool to actually see something that you've been dreaming up in your mind for the last 6, 7, 8 years come to fruition, but we had a great team and we made it happen. So then it came time to transport this thing down to State Line Launch Ramp at Lake Powell. I didn't get to see this firsthand, but I saw some pictures and it looked like quite the sight out on the highway. So the dip tank was finally installed at the State Line Launch Ramp on May 1st. We've been using it ever since. We've used it to perform over 400 decontaminations so far. The boating public has been very appreciative and supportive of the system. A lot of them have actively kind of, they seek out the dip tank as their preferred method of decontamination. Part of it's just kind of, it's cool. It's got the new wow factor, I guess, if you want to call it that. But it also, and I'll get to this in a slide or two, but it also kind of simplifies the process, and I think that puts a lot of their minds at ease. I think our program staff have been very grateful for this new tool in their tool belt as well. Obviously, climbing under under some of these wakeboard boats and things to do a decontamination when it's 110 degrees out and 140 degrees on the asphalt. That's not the greatest job. With this system, they no longer have to do that. We typically will plan to operate the dip tank roughly from early May through September each season. Part of that will be dependent on boat traffic, but we're going to be focusing a lot on those complex watercraft like those wakeboard boats, maybe some small cabin cruisers, the ones that we really start seeing in large numbers come, mid to late June through the end of August. So while this has been a huge tool, it certainly hasn't been all unicorns and rainbows and everything has been smooth. We've definitely figured out a few things that we need to consider. So considerations, like I said, we've done 400 dip tank decontaminations to date, roughly. We have found that this has resulted in a significant reduction in the pressure on our wall weep semi-permanent decontamination unit. We primarily use that unit now to tackle some of the general recreation boats, the fishing boats, the PWCs that are fairly simple to do, and we can crank those out pretty routinely, pretty quickly. The wakeboard boats all go to the dip tank, and that seems to be a really nice balance between those two systems. It's also been a huge boon in customer service. We've reduced those wait times for people, so they're no longer having to wait hour and a half, 2 hours, 2.5 hours for a decontamination. There's also kind of this perception that there's less meddling with my boat, so to speak. Sometimes people get nervous, and I probably would too if I had a $200,000 boat. When you start trying to figure out where attachments go underneath the boat and trying to pump in hot water to it. There's none of that, so they can simply operate their boat just like they did if they had just launched it at the reservoir. And then also, I think a big thing has been the impression that we are actually trying to improve the boater experience. We're trying to address some of the things that the public has expressed concern or frustration about. We're trying to invent new things that will not only help us in our program, but also create a more seamless and convenient experience for them. So some of the challenges that we have seen that we're going to have to address in version 2.0, which I'll talk about in a second. Lake Powell has a bunch of sand around it. It's a very sandy environment, very windy at times, frequent wind. And we found that a lot of times that wind is blowing some of that sand from those nearby areas into the area around the tank, or sometimes even into the tank. That's obviously something we're going to have to address as we move forward and we start talking about maybe potentially putting in another dip tank at Lake Powell. We originally used 3-inch diameter shade balls to reduce that evaporation and maintain some of that water quality. 3 inches has proven to be a little bit small for some of the boats and trailers going in and out, and they've been getting wedged into some of these areas. So for the next iteration, we're talking about larger shade balls, probably 4-inch or potentially even 5 inches in diameter. With the number of boats and trailers going into and out of the tank, we also saw that a lot of them are tracking gravel into the tank. That's required us to go in and clean the tank out every 4 to 6 weeks. So we're going to have to figure out a way to mitigate that as we talk about the next version as well. And then probably lastly, but probably the most significant thing that we've encountered is we're going to need to modify the approach angle into the tank, because one thing that we figured out fairly soon right after we opened it was, depending on the configuration of the vehicle, the tow vehicle, the boat, the trailer, the hitch height, whether there was a spare tire under the boat trailer, all these things, we found that sometimes there'd be an occasional boat where they couldn't realistically back down into the tank without catching the lip of the trailer right on kind of that transition plate going down into the tank. So we had to make some on-the-fly adjustments. Jonas and Garrett were huge. Clem, our interdiction specialist down there, they put a lot of time and effort in trying to figure it out. We came up with a good game plan. So we can now accommodate roughly 95% of the boats going into and out of the tank. But I think this is something we can address going forward from the outset for this next one. And future expansion. So we have the one down at State Line Launch Ramp at Lake Powell. We're talking about the potential of putting in one at Bullfrog this next season, and then probably another one up along the Wasatch Front near Salt Lake, either this coming season or the following season. So you may be asking yourself, can I get one of these? I will throw a few questions back at you. One, do you perform enough decons at that location? Obviously, if you're performing 100 decons at that location, it's not going to make sense for you. Kind of along with that, do you have enough wakeboard boat traffic? These are built for wakeboard boats and complex boats to fill all those systems simultaneously. If you're just decontaminating fishing boats, the investment probably isn't worth it. What are your desired seasons of operation? Like I said, we're planning on operating this from late April, early May through September. We have a long boating season at Lake Powell. If you have a short season and you're only worried about 2 months, again, the investment may not be worth it. Space utility requirements, and what is it going to take to maintain that water temperature at 110 degrees? Lake Powell gets super hot in the summer, so we don't have to worry about that as much as you might if you're at a high alpine lake and you seldom see anything over 80 degrees. And like I mentioned, can you provide both the dip tank and another decon method at the same time so you don't have the dip tank being taken up by fishing boats as wakeboard boats have to wait? Oh yeah, you're probably saying, what's the cost? Total cost, $800,000 to have it fabricated, delivered, and installed. Seasonal propane costs, we're estimating at around $7,500, and we did have to replace one generator at the cost of $4,000. So many of you have probably already seen this, but for those who may have not so far, here's another glimpse of this new tool.
So Nate, you have about 3 minutes left.
So with that, I would just like to thank National Park Service, Bureau of Reclamation, Arizona Game and Fish for our partnerships at Lake Powell, Fish and Wildlife Service for their financial support of our containment work there, Garrett and Jonas of Clean Wave for being unbelievable problem solvers, Clem Wasachek, one of our interdiction specialists at Lake Powell, who is putting his soul into this thing and making it work, and then our great team of interdiction specialists around the state that make it possible for me to proudly present on our program. So with that, thank you very much. I'd be happy to take any questions.
Excellent, Nate. We have, I think, a moment here for a question. Okay, I guess to just verbalize. So you did have an attendee want to touch base with you about actually materials that you can spray on the boat to minimize adhesion, and I will share that information with you after the webinar. So thank you, Brett Orsak, for that comment. And then Nick Zerflu is asking, do you have a sense for what the boater resonance is in using the dip tank? Are most of them Utah residents, or are they coming, from a regional area? Are you capturing any of that information of? No.
I'm sure our team down at Lake Powell could probably speak to that more. As far as I know, I wouldn't say anyone is more prone to use the dip tank than anyone else. I think typically we see the same people asking for decontaminations coming out of Lake Powell. So if they're routinely asking for a decontamination, I think anyone with a wakeboard boat who is coming off one of those two ramps is likely to see the dip tank and then ask for one.
Okay, and then real quick, I have a question from John Wolschlager. So did you say the temperature is 110 degrees Fahrenheit? Is that not cooler than the recommended decon temperature in the past? And are you confident that's effective?
Good question, John. So yes, we went through this thoroughly. It is 110 degrees Fahrenheit. If you look at the mortality table, at 110 degrees Fahrenheit, I believe you achieve 100% mortality at like 48 seconds. The exposure time in this tank is probably closer to 5 or 6 minutes. So we far exceed the exposure time required for 100% mortality. And I would also like to point out, we are ensuring 100% mortality because we are completely filling all of these systems. Whereas right now, without a dip tank, we're filling maybe a half, if we're lucky, of some of these systems. So we're kind of relying on an assumption that half a ballast tank filled with hot water, the heat will just generate itself and kill anything else that may be in there. We don't have to worry about that with this system.
Excellent. Okay. That looks like that actually addressed one of our other attendees' questions. So, but we have to stay on schedule here, Nate. So thank you so much for your time and your presentation. I'm going to hand it back to Tom Wolf here to introduce our next speaker. So thank you.
Nate, great work, Team Utah. Good job down there. Our next speaker is Sherry Percelli. She's a biologist with Bureau of Reclamation Technical Services Center in Denver, Colorado. She's done a lot of work on invasive mussel eradication and control methods. They do great work down there at Reclamation in Denver, and she'll be talking to us today about innovative mussel biological control methods. Take it away, Sherry.
Great. Thank you so much. Can you see my presentation?
Yes, it looks great, and I can see you.
Okay. Oh, perfect. Good. Okay, great, wonderful, thanks. So again, my name is Sherry Pucciarelli. I work for the Bureau of Reclamation as a biologist. I'm also the Invasive Species Research Coordinator for our Science and Technology program. Today I'm going to be presenting on 3 invasive mussel biocontrol projects that Reclamation is currently funding. So at Reclamation, our Science and Technology program has invested in invasive mussel research for many years now. Since 2010, the program's funded over 100 projects. Currently, we have 28 research projects in progress. And a lot of this research in the past has focused on developing methods to control mussels at hydropower plants and also to develop early detection and monitoring methods. But more recently, we have been funding projects that are focused on trying to develop methods to control Disseminated mussels in large bodies of water, which we call open water control. So as you're very well aware, it's very difficult to control mussels in a large body of water. There have been some successful efforts, but they were in small closed systems. And in those cases, eradication relied on potash or copper compounds. But when you're talking about a reservoir the size of, say, Lake Mead, you run into a lot of challenges: scalability of these methods, cost, trying always to make sure that you are using something that's species specific and that there's minimal impact to the environment. So with our projects, we're trying to kind of overcome these challenges. And so we're focusing on biological control methods. So we have one project that's more focused on traditional biocontrol using natural enemies, just parasites or predators from the mussels or from the native range to be used in the invasive range for control. We're also looking into genetic biocontrol methods where you use emerging molecular techniques to alter the genome to achieve control. So our first project that I wanted to discuss today is more of a traditional biological control project. Jackie Keel is the Reclamation project manager, and we are collaborating with Dan Malloy of Malloy and Associates on this project to search for Disseminated mussel parasites in Eurasia that can hopefully cause quagga and zebra mussel mortality. So this is a little bit different than maybe a traditional biological control project because we're not looking for parasites in zebra and quagga mussels. Dan actually already spent decades surveying parasites in quagga and zebra mussels in their native European ranges, which is kind of outlined here on this map. And he didn't find any parasite with the potential to be a biological control agent. So the focus has shifted to looking for parasites from closely related Dracynid species, or what he calls cousin Dracynid species. The idea here is that zebra mussels and quagga mussels might be naive to these parasites. They've never been exposed to them, and it might cause more mortality. So the search area has shifted. Outlined here on this map. They're examining Dracynid species in the Balkans and Turkey, species like Dracaena anatolica and Dracaena carinata. So some of the potential advantages of this project would be that, if they find a biocontrol agent, it would only need to be applied to a small part of the water body. It would be self-perpetuating, self-spreading. Parasites are the target here because they're typically the most host-specific of natural enemies. And there are many well-documented cases of animal populations being decimated when exposed to a parasite from a closely related species, especially in cases when they've been geographically separated for a long time. So an example would be the eastern oyster populations in the United States were quite abundant until the 1950s, when a parasite from the Pacific oyster came in and killed about 95% of eastern oysters. So this has happened before. So Dan has done a great job of developing a network of collaborators across the world. He has 18 collaborators for this project from 9 different countries, and their contributions have really accelerated the progress of this research. We have people that are helping to identify the populations, people helping get collection permits, actually going out collecting and doing dissections. And we also actually just got another collaborator from the Army Corps of Engineers that has a lot of experience with biological control projects. So they're going to be helpful in guiding the project. And then Reclamation is assisting with genetic identification of the parasites. So for this project so far, they have collected and identified parasites from over 5,000 mussels in 4 different species from 5 Eurasian countries. They've done collections in Albania, Bulgaria, Montenegro, North Macedonia, and Turkey. Dan has also been able to establish a field laboratory in Montenegro, which you see on this picture here on the left. This is on a property that has a spring water source, so that's available year-round to feed these 72 custom-made aquarium for transinfection studies. Picture there on the right. And he has a team of scientists working at this lab year-round to keep the study going, even when he's not there. So Dan has demonstrated actually his hypothesis that parasitic infections can be transmitted from these cousin species into quagga and zebra mussels. And this was the first time I think that this has ever been demonstrated in Dracynid mussels. He did a lab study where he exposed zebra and quagga mussels to Dracaena carinata, and they found that the 5 Dracaena carinata ciliate parasites were actually transinfected into quagga and zebra mussels. They did a study, a field study at Sasko Lake in Montenegro. They were able to get permits to do this study. Zebra mussels are actually invasive in this lake, and they were able to expose native Dracaena carinata to those zebra mussels. And they found that the parasites were transmitted into the zebra mussels. So, this is a great proof of concept. Unfortunately, those parasites that they were testing in this study did not appear to cause much mortality in the zebra and quagga mussels. So they're probably not a biological control candidate, but they do have the infrastructure now set up to continue doing these studies with other parasites. So they have an upcoming trip here in a couple weeks, October 1st through 10th, where they're traveling through north central Turkey to find additional Dracynid populations to use in upcoming transinfection trials. Right, so now I will kind of shift gears to more of a genetic biocontrol project that we have going on. Yale Pasamanik is the reclamation researcher responsible for this work, and he actually just recently successfully sequenced the Quagga mussel genome. He partnered with a collaborator at the University of Alabama to do this work, and this is a really critical step in approaching genetic biocontrol. If you don't have the genome of the organism that you're trying to target, you don't have those gene targets needed. So this is a really big step. For this project, they collected quagga mussels from Lake Mojave at Davis Dam, and they sequenced, or they isolated genomic data from one individual for sequencing. Just to basically kind of go over the process, they completed DNA sequencing, which produced millions of sequence fragments. They used two complementary sequencing technologies, Illumina HiSeq and PacBioSequel. And then they created a config assembly, which is basically a bioinformatic analysis used to reconstruct the genome. And that produced about just under 9,000 contigs. Next, they did HiSeq scaffolding, which created 16 scaffolds that match the 16 chromosomes in the quagga mussel's genome. So in the end, the genome is about 1.6 billion base pairs, which is about half the size of the human genome. So it's kind of a visual representation of what I just described there in the process. They actually had to do this 100 times over to get a complete construction. So after they did this, they did an analysis, a BUSCO analysis, which looks at the 11 evolutionary conserved genes. And they found that more than 96% of the expected genes were found. And so this is actually one of the best bivalve genomes that are currently available. This graph here shows the bivalve genomes that have been sequenced. The second line is the Yale's quagga mussel genome. And this is the BUSCO score, but basically the blue portion is the complete, and the red portion there is the missing percentage. So this is, as you can see, compared to these others, this is a very complete genome. They did a really great job. So Yale is working on annotating the genome now, trying to identify targets for genetic biocontrol. Typically, these targets would be genes associated with reproduction, including gonad and gamete development. But actually, the information from this genome is already being used for another genetic biocontrol project that Reclamation is funding, and I'll move on to that project next. This project came out of a prize competition that Reclamation ran in 2018, which was part of the Reclamation's commitments to the Department of Interior's Safeguarding the West initiative. And if you're not familiar with a prize competition, they're basically just a tool used to accelerate research on challenging topics. They incentivize the private sector and citizen solvers using cash prizes. And for our prize competition, we were offering a $100,000 cash prize. Our objective was to seek theoretical, innovative solutions to control mussel populations in large bodies of water. And so our full prize winners were Steve Sir and Marie Cloud Snoot, the co-founders of a lab in Michigan called BiomeLab. And their solution was a genetic control option. It was to create an engineered disseminated neoplasia, which is actually a form of naturally occurring cancer that does occur in marine bivalves and some other animals. It's quite rare, and it's unique in that the cells are spread by direct transfer from one individual to another. So it's a form of contagious cancer. So the idea would be that a single individual could pass these cells throughout the population, leading to population collapse, which has happened in some marine bivalves. So the goal of this project would be to replicate this neoplasia in quagga and zebra mussels. And so this was a theoretical idea. Reclamation established a cooperative agreement with BiomeLab to continue this research, and they've actually been working on this research project for about 2 years now. So the short-term objectives of this project were to develop methods to culture adult and embryonic cells, genetically modify muscle cells to induce cancer, and introduce these engineered cancer cells into live muscles. And this in itself is a lot of work. You know, they're working with a novel system here. This has never been done for Dreisseniid mussels. So a lot of work needs to go into understanding these basic foundations. Some of the long-term objectives would be to select for lines that efficiently disseminate and produce lethality, and then test, I guess many years would go to testing for off-target effects and efficacy under controlled laboratory and in real-world conditions. So some of the accomplishments that BioMe Lab has achieved so far. They have developed methods for maintenance of adult quagga muscle cells and tissues in culture for greater than 4-week time period. So you can see pictures up here of gill cells in culture and then mantle cells. And these mantle cells are actually producing calcium carbonate structures like they would be when they're creating a shell. Kind of interesting. They've also been able to successfully achieve in vitro fertilization and growth of muscle embryos. Those are pictures of muscle embryos here at the bottom. They've also tested a variety of methods to confirm efficacy of gene transfer. So they've successfully introduced fluorescent reagents and microbeads into live quagga muscles. And you can see those here in the gills and mantle in these pictures. And as I mentioned, they are using Yale's sequenced genome, and they used it to characterize gene targets. They focused on the P53 genes. These genes regulate cell cycle, and they can attempt to knock out these genes to achieve cell transformation. They've also produced tools for genetic modification and cell transformation. So they've developed more than 12 vectors that can deliver transgenes or CRISPR-Cas9 components. And while they were, learning to culture the muscle cells, they tested the functionality of these quagga muscle-based vector constructs and transgene delivery methods using human hex cells and fly S2 cells. And they do this using reporter genes to test the functionality. They tested GFP, M-cherry, and firefly luciferase. These pictures here, I believe, show M-cherry in reporter genes in HEC and S2 cells. And this just allows for tracking of expression of the introduced genetic material. And finally, they have also been able to establish methods for microinjection of quagga muscle zygotes. Picture of the zygote here, and then they're actually are using microinjection to inject genetic material there. So this is a big step forward that just recently occurred. So, overall, we're at Reclamation, we're very excited to be part of all of these projects. They are long-term research projects. There is the possibility that they might not found to be safe or effective, but we think that it is worth pursuing these projects because they are developing some really important methods that can be used for a variety of biological control projects in the future if these are not found to be successful. So we're pretty excited about this work. And on the screen, I have my contact information, Jackie's contact information, and Yale's. Please reach out to us if you have any questions after today. And if I have time, I'd be happy to answer any questions.
Awesome, Sherry. Thank you so much. I do have a few questions for you already. So let me verbalize these to you. We'll see where we go. Okay, so Kat De Rivera asks, what native riverine and estuarine bivalve species will be in experiments about vulnerability to the parasite that successfully infects zebra and quagga mussels?
I don't have a list right now. Obviously, we're still in the searching phase, but it will be a very extensive list, just like most biocontrol screening projects. We'll definitely follow all of the requirements. Obviously, all closely related species will be the primary organisms tested, and then I think it'll expand from there. It's pretty extensive when you do these studies for biological control. A lot of species need to be tested, and it takes years.
Right. Yeah. Okay, so let's see. Bear with me. This feels like a long question. Maybe I'll ask it in pieces. Okay, so Teresa Thome asks, how did the genome from the single specimen from Lake Mojave differ from the genome sequenced at the Danube River in Austria?
I believe, as far as I know, it's definitely more complete. And I know Yale knows about that one, and it'll probably be, part of when he's doing this annotation, looking at that a bit. As far as all I can say about it is I know it was just a more complete genome in the end.
Okay, sort of to follow the rest of the words that Teresa asked, was curious how different the populations might be. The genome work in 2019 from Calcino et al. Was focused on quagga mussel freshwater tolerance that may have unique applications for quagga mussel control. It was kind of secondary to that. Okay.
I don't know much about that other genome, but that might be a good question for Yale if you'd want to reach out to him to get additional information.
Okay. All right, so those are the two questions I see now. Let me just double check someone hasn't put something also in the chat box. All right, okay. If there are any more questions for Sherry, please pop those in the question box before we move on to our next speaker. I want to make sure we give time. We do have a couple more minutes, but if there are no more questions, okay, I am not seeing any more come in. So thank you so much, Sherry, for your presentation. This is all fantastic work. I'm curious to see where it all goes. So excellent. Thank you. Back to you, Tom.
Thank you, Sherry. And as a manager, one of the questions we always get is, what do you do if you get these things? And so the work you guys are doing with Reclamation down there is outstanding. It's critical to help. Having tools in the toolbox to address these things is an important step if in fact something slips through our prevention measures. So thank you. Our next speaker is Megan Winder. Megan's a statistics PhD student at Montana State University. And Megan, welcome. I haven't had a chance to meet you yet because we're both in the same state, but I hope to following this presentation sometime. She's doing her research on statistical applications in ecology and environmental sciences, and her presentation today is evaluation of dreissenid mussel detection probabilities using Bayesian hierarchical occupancy models. Thank you, Megan. Please take it away.
Okay, great. Can everybody see my presentation?
It looks great. The only thing, Megan, I know you're wearing those blue light glasses, but as long as you can see. Awesome.
Yeah, I can see this fine.
Great. Okay. Thank you.
Okay. So my name is Megan, and as Tom said, I am a PhD student at Montana State University, and I am a statistics PhD student, but I'm really interested in ecological and sort of environmental science applications of statistics. So he read the sort of formal title for my talk, but the more informal title would be how many plankton toast samples is enough to take to really understand if we're capturing zebra and quagga mussels if they're present in a water body. Okay, so I'm not going to spend a bunch of time talking about how bad dry skin mussels are, or why early detection is really important, because most of the people in this talk are probably foremost experts on those types of things. So it's maybe a waste of my time to tell you about how bad they are. So we'll just start with an overview of how plankton toe samples work, and just a reminder about how those samples are sort of grouped together to make the data that we received. So you're probably familiar, there's a plankton toe net driven around behind a boat, and then usually there are multiple toes taken from a lake on a given day. And then those samples are all collected into sort of one aggregate sample, and that's what we'll call a sample moving forward. So one sample is about 5. It's usually 5. Sometimes it's more, sometimes it's less. Typically 5 plankton toe aggregate samples. So maybe confusing, but one sample moving forward is 5 or so aggregate toes. And then those samples are taken back to the lab and analyzed with a microscope, and sometimes followed up with PCR. So we'll just talk about some example data that you might observe from plankton toe samples. So let's say we go to a lake and we're going to take 5 samples. So again, 5 of the aggregate samples from before, so about 20 to 25 toes. And let's say that these are the data that we observed. So the little red in the jar means that we didn't observe any dry skin mussels in each of those samples. So if these are the data that we observe, what can we sort of learn from these data? Like, do we think that the lake has zebra mussels in it? Do we think that it doesn't? Are we unsure? Should we collect more samples? So I'll just give you a couple seconds to think about that and sort of evaluate what we would want to do moving forward from here. Okay, so obligatory, like, awkward silence, but I think that's maybe enough time to say that, like, okay, we took these 5 samples, but we don't really know if dry skin mussels are present in the lake or not. So let's take another 5 samples. Okay, and from these 5 samples, we observe 3 more that we don't detect dry skin mussels in, but we observe 2 where we do detect them. So now we know that zebra mussels or quagga mussels are present in this lake, and we have some information about how detectable they are in the lake. And so if we were just going to take a guess at the detection probability based on these samples here, we would guess that the detection probability is somewhere around 20% for occupied lakes for zebra mussels. So let's do another example. So we take 5 samples, they're all negative. So we don't know if zebra mussels or quagga mussels are present in the lake. So we take another 5 samples and they're also negative. And so at some point we can become more confident the more samples we take. So we take another 5. The more samples we take, that zebra mussels or quagga mussels are not present in this lake. But the question that we're sort of looking to answer here is, how many samples do we need to take for there to be enough zeros for us to be pretty sure that this second lake is not occupied? So should we keep taking samples, or is 20 about enough? So this sort of leads into the research questions. This is more, I guess, formal than the last sort of slide with the diagram, but again, we're really interested in that detection probability. So how many of those jars are green? But it's a little bit more complicated than that, because we can include other factors. So this is the second question, but we can include other factors about the water body to to better inform those detection probabilities. So things like water temperature might make sense, water body size, things like that. And then given that detection probability, how many samples should we be taking to be sure that we're capturing dry sand mussels if they're present in the lake in at least one of those samples that we take? And then how does that compare to the sampling effort that is currently being done? So these are the data that we have for this exploration. They come from Bureau of Reclamation. There are data on 288 lakes in the western part of the United States. On this map, you can see that there are more than 288 dots, but all of the points that are close together. So these ones up here, you can feel pretty confident that they're from the same lake. So that's why there are more than 288 points. And then we split the data into seasons, which are water years. We use the USGS definition of water year to separate these data into seasons, which we won't talk about a lot here, but it is important if you think more about the model. So October 1st of a given year through September 30th of the following year. But if you're choosing to ignore the model, which is totally fine, then you can ignore the seasons as well. So again, here's the model. If you're not interested in statistics or you don't care about the model, that's totally fine. I just thought I would throw it up here in case people have questions about it. But this is the model that we fit, and all we really care about in this case is the blue P right here. So that's the detection probability. Everything else you can ignore, or if you have questions on it, I'm happy to take those after. So first, we fit a model where we just estimated the occupancy probability and the detection probability as constant, which we'll talk about that in a minute, but probably doesn't make sense to do, but at least gives us some basic understanding of what the occupancy probability and the detection probabilities are about, where they're about centered. So the occupancy probability is estimated to be about 5.2 to 8.2%, meaning that about 5 to 8% of the lakes in the data set are estimated to be occupied with zebra mussels or quagga mussels. And the detection probability, so that's estimated to be around maybe about 60%, but around 54% to 63%. So about 60% or so. But so that detection probability is conditional on the lake being occupied. So there's this assumption in the model that we fit previously that you don't detect dry sand mussels if they're not there. So that's the false positive assumption for that model, which I think makes sense if you think about it moving forward. So the next question is, can we include some environmental factors to help better model the detection probability, and really it makes sense to do occupancy probability as well, but we'll just go one at a time here, and we'll just include water body size for detection probability. And you can see, if you pay attention to the occupancy probabilities here, you'll see how those change a little bit as we include water body size for detection probability. So we didn't, occupancy probability is still constant across all of the lakes in the data set. So we didn't actually change anything for that part of the model, but these two probabilities kind of work together to inform each other. So when you change one or you add something to one of them, so water body size is modeling the detection probability, so it has nothing to do with occupancy probability, but they sort of inform each other. So as one changes, the other one will change a little bit too. So now the estimated occupancy probability is a little bit higher than the last model, but not too much higher. And then you can see the estimated detection probabilities for each of these water body sizes as we move across the x-axis of this plot. For streams, the estimated detection probability is around 0.8 or so, and then as low as for large water bodies, about 10%. So this is just highlighting that, again, the previous model with constant detection probability is probably not appropriate, because as soon as we put in this variable for water body size, we're getting huge differences in the estimates for detection probability for different water body sizes. Okay, so moving forward, so how many samples should we take? Again, reminding you that one sample is 5 or so aggregate toes. So if we look at this plot, X is the detection probability, or on the x-axis is detection probability, and the number of samples is on the y-axis. So if we think back to the model with the constant detection probability, the estimate for that was around like 50 to 60%. So for each lake in a given year, you should take around 4 or 5 samples. Okay, and so that's fine. But now let's think about the estimate for the detection probability for the large lake. So that was around 10%. So based on this, we should be taking around 29 samples in a lake per year, which is a lot more than 5, certainly. And especially, so yeah, for streams, the detection probability was around 90. So if that's the case, then you should really be taking like 2 samples. It would be enough to have a 95% probability of detecting zebra or quagga mussels if they're present. So you can see based on this table that the number of samples really depends on the detection probability. So moving forward, we would really like to include other covariates in this model. So water temperature, I think, would be really important for detection probability, because you're more likely to detect veligers when they're spawning or close to a spawning period, which occurs at certain water temperatures, as you're probably all familiar with. And then pH and dissolved calcium, there's sort of a threshold for or like a sweet spot in pH and dissolved calcium where dry sand mussels habitat the lake. So I think that those would be really helpful things to include in this model. However, these values are not collected, or I don't have access to them with the data that I have. So we have pulled them from the water quality portal, but there's some mismatch in dates and space and things like that. So I think that if these were collected with the data, that would certainly be very helpful and something that we could think about moving forward. We're continue to work on this in the future, and so you can keep an eye out on a paper which will include these covariates, but for now we've just included water body size for this talk. And then on the plot on the right, this is a histogram of the number of samples per lake per season. So back to this table, like how many samples should we be taking, but really how does that relate to the number of samples that we're currently taking? So anywhere from 1 to 104 samples are taken at a lake during a given season, with a median of 5. So about half of the lakes have 5 or fewer samples taken per year. So if the detection probability is low, like below 50%, really we should be taking more than 5 samples, because everything here below 50% has more than 50 samples or more than 5 samples that you should be taking in those scenarios. And then additionally, I think, I don't know what else I was gonna say there. Okay, Yes, so big thank you to Sherry at Bureau of Reclamation and Yale at Bureau of Reclamation and Bureau of Reclamation as a whole for funding this project, and Sherry and Yale for helping gather the data and answer all my questions about the data, how it was collected, things like that. And then here's some references.
Excellent. Thank you, Megan. Let's see. All right. Do we have any questions for Megan? Okay, let's see. I do have 2 questions for you. Let's see. Okay, Allison Zach asks, when are the results or publication of the results of this research to be expected? And then how can or who should we contact to get those results? So what are you thinking on timeline?
Yeah, so for the timeline for that, I would say we're planning on finishing up a draft of the manuscript in the next month or so. So after it works its way through the publication process would be when we could expect that.
So optimistically within a year.
Yeah.
Okay, awesome. Okay, I think it's a clarification from James Dominguez asks, regarding the samples per year, I think I'm understanding this correctly. Are these standard routine sample locations or randomized locations? Does that maybe make a difference in interpreting that information?
Yeah, so the samples that we have that we're analyzing for these data were all the routine locations
Okay.
for the samples. So they were not randomized. So we're just assuming that you're taking samples from the lake, and the lake, there's a constant detection probability and a constant occupancy probability over the entire lake. So if that's not the case, that would be when the location within a lake would matter.
Okay. I have another question here from Nate Owens. Okay, so bear with me because I have to read a bunch here. So wait, hold on, let me pop this out so I can, okay. So this study focused on the number of samples taken, where in the field the volume of water sampled per plankton tow can vary greatly. Is there a way to translate these conclusions into volume sampled instead of number of samples? And is that something that you have considered?
That's a really good question. I am not sure that I have data available for all of the volumes of water for all of the observations, but this analysis definitely could be run on a subset of the data where the volume of water was available. And I think that that's really interesting and highlights a great point that I hadn't thought of as a statistician, but it definitely makes sense now that you say that.
Okay, so maybe at the very last part of his question, maybe then clarifies. So then he was guessing, did this study assume a constant volume of X? Is that part of the assumptions in that it was a constant volume or not?
Yeah, so it would assume that each of the samples, that the sampling effort is sort of the same, which is maybe not a reasonable assumption, because some of the samples have like 5 or so aggregate tows, but some of them are just 1 tow. And so the sampling effort is maybe not the same for each of those.
Okay. And I have, looks like, well, maybe it's not the last question. If someone puts another one here, I have another question from Adam Sepulveda. So from a statistics perspective, what do ideal or study design look like? Sorry, ideal data. Did I not say that word? What do ideal data or study design look like?
Yeah, so the ideal data, you can continue to collect these samples like you are. It would be helpful if the number of toes was always recorded, or the volume of water sampled was always recorded, or both of those things, because then you could incorporate the sampling effort. But like I said, those weren't always incorporated. And then additionally, when the toes were collected, if pH, dissolved calcium, water temperature were also collected.
Okay, take note, everybody. All right, well, let's see. I do not see any more questions. Oh, no, that's not true. I've got one more that's just come in. Okay, Nick Zerflu asks, most Western state protocols call for sample locations to be where mussels are likely to be introduced or congregate. So boat ramps, marinas, dams, etc. Does the model take into account sample location within the water body?
No, so that was one thing that we wanted to include in the model, but because the toes are aggregated, the number of samples within each location in a water body, so say at like Holter Lake in Montana, you're taking, you have 3 different sample sites, but you only took one aggregate sample at each of those sites in a year, then there's no replication there, and you need to have the replication to inform the detection probabilities and the occupancy probabilities. So we had to collapse it back to the lake level. But that's a great question. I really like that question.
Okay, excellent. Well, I do not see any more chats or questions for you. So thank you so much for sharing your work with us today, Megan. Appreciate it. Good luck to you in finishing that up. Okay, so I guess it's back to you, Tom.
Thank you, Megan. Any information that can help inform our sampling efforts is really appreciated. We always go by the guidance of more samples is better, which I think is always the case, but how many is a question that the more information and research that helps support that, the better. Thank you. Our next speaker is Cody Youngbull. Cody is a research professor at the University of Montana Flathead Lake Biological Station. He's done some great work to transfer technology from the medical field into early detection technology for dreissenids. Been privileged to work with him at the biostation and up in Flathead Lake. His talk today is continuous on-site quantification of invasive mussel eDNA at Inks Dam National Fish Hatchery. Cody, please proceed.
Hello, hey. Somebody confirm that you can see my presentation now. I'm using Google Slides. And
Confirm.
Can you see it?
That's good.
We got it.
Okay, great. Thank you all. I've enjoyed listening to everyone. I think that my presentation, obviously, it dovetails really with everyone we've just heard from. Yeah, we've got Nate's struggles and successes at delivering instrumentation. I feel his pain in developing instrumentation for the field. And I stand on the shoulders of giants with Sherry's presentation, the biomolecular detection behind all of this. And then Megan, I was really interested in the statistics of sampling. This really puts that on its ear. It's a different way of sampling, a different way of interacting with the environment because it's continuous monitoring. It's like a smoke alarm for eDNA. And so it can really change the way we monitor the environment. It's been a long time in coming, this instrument. I've been working on it now for, 11 years when we first started this instrument, mostly funded and developed at the Flathead Lake Biological Station. But recently it's been licensed by a company, NS2. You should go to ns2co.com and see the instrument that they're going to be putting out commercially soon. And recently we've got a lot of funding and support from U.S. Fish and Wildlife Service, NOAA, and the National Science Foundation to help develop this instrument. So without further ado, let me tell you what what this talk's really about is deploying this instrument to Inks Dam National Fish Hatchery down in Texas. This is an aerial view of the hatchery. What they do out here is they produce most of the catfish, the channel catfish for the tribal fisheries management in the West. And so they're shipping catfish out all over the West. And recently, this lake, which supplies their water, has come up positive for zebra mussel. And so there's great concern that this might be a vector for disseminating invasive species all over the West, and so they're very careful about that. And they have mitigation plans, and they're now not taking their water from the lake. In some situations, they're taking it from well water. But even then, there's questions about can there be mussels surviving and getting through and being spread throughout the West. So they're very interested in running lots of sampling in their various processes, lots of biodetection, I mean to say, in their various processes. But that can be very cost prohibitive, and also the time delay and waiting to get those samples back can be a big problem for them. So they really need an on-site detection tool, and it needs to be cost effective and amenable to how they work. And so in comes this tool that we've been developing for years at the biological station, initially with the large support from the National Science Foundation. This is a picture of it, briefcase-sized tool by the side of the lake. And this is an example of some of the data it produces. These are, I'll go into the details of how this data is produced, but the red dots, if you could see them, are individual detection events that did not have zebra mussel DNA in it. And the green dots are individual detection events that did have zebra mussel. This is a test sample where we spiked environmental water to make sure the machine is working. But this is the way the raw data looks. And this gives you a sense of scale. That little briefcase, it can fit in the overhead compartment of an airplane or the seat in front of you, and it does travel and can be held like a briefcase. And so this is one of the amazing things we did with it, funded by the U.S. Fish and Wildlife Service. We took it to Lake Mead. We put it on a boat, and in traveling from position 1 to position 7, we measured, sampled the water and detected quagga mussel at those locations. I think most interesting, you can see at position 5, we're detecting about 3 gene copies of zebra mussel per microliter of lake water. And that's about 10 kilometers from any known observed mussel beds. So there's eDNA everywhere in this water. It's everywhere. This is a particularly contaminated lake, but no matter where you are at, there's eDNA in the water. And this was done on the boat. And one of the interesting things to point out, though, is we're looking at a type of eDNA called free eDNA in this particular case. The machine can, so something that people should realize is that not all eDNA is created equal. There's different types. There's cell-associated, particle-associated, and free. But when cells in the veligers pop, they release their contents, and that internal DNA is distributed throughout the water. It's consumed rapidly by background microbes, and the sunlight breaks it down. But if it's not associated with the cell itself or the particles, it's called free eDNA. It's greatly understudied because people don't have these kinds of tools to go out there and measure in situ, because it's hard to stabilize. It passes through filters, and it doesn't last very long, as I say. So this is one of the unique things you can do with an on-site mobile detection tool for eDNA. Here's another one. This is on-site validation of biodecontamination efforts. Here we're collecting water off of this boat prior to decontamination. You can see we're getting lots of hits for the zebra mussel eDNA, and this is quagga mussel eDNA, excuse me. And then after the decontamination efforts, we get no hits. And the machine isn't occupied like standard PCR machines, so we don't disrupt traffic flow. It still takes the same amount of time and still doing PCR, but so it takes about an hour. It's an hour delayed, but you can introduce new samples every few minutes into the instrument and just process them continuously. So this is another unique thing that you can do with an on-site continuous detection tool. So the reason the data doesn't look like standard PCR curves is because it's operating based on digital PCR. In digital PCR, you start with your sample, which contains your target nucleic acids, and you partition it into thousands or tens of thousands of nanoliter droplets, and then you do your PCR. So what you're doing is you're doing tens of thousands of presence and absence detections, and you're bootstrapping using the statistics of that to quantify the original starting concentration. So this is actually a form of quantitative PCR, droplet digital PCR, and because of this, it's robust against inhibitors. I'll give you an example. This is what the data looks like. This again is spiked environmental water. Here with no DNA, target DNA in it, and on the left with target DNA. If there's inhibitors in the water, gasoline in the sample, humics in the water, the amplification efficiency goes down, so the height of your ones will decrease. But so long as you can count the zeros and count the ones, you're still able to quantify. So you no longer need references. You can still run controls to make sure the machine's not contaminated and that it's working, but there's no need for a reference or comparing it. So it's absolute tighter measurement of the extant eDNA in that sample. This is a still photo. I'm going to go to a video, but the way we form those individual, the way we partition the sample into droplets is we use a very special oil that doesn't mix with the water, so there's forming emulsion. And when you bring in that water that has the primer probes and the master mix, it breaks it into tens of thousands of individual reactions. So let's watch a video of that. Here's a video of it happening in slow motion. And the blue is the water droplets that are being formed. Each one's about a nanoliter that has the reaction in it. And then you run them through a thermocycler, and you amplify that DNA, and you get the data like I was showing back here. If it amplifies, it's very bright, and if it doesn't, it stays dim. And so the machine can absolutely work on pure DNA, as I said, free DNA, but it can also, you can walk up to it with any prepared sample. So this is the typical process of what happens after you get the water sample, or you filter it, which is this step is a concentrating step, and then you break down the cells and you leave behind the internal components, separating that proteins from the nucleic acids, and then you put the nucleic acids in a PCR machine. But in the past, decade or so, there's been great development in types of enzymes, lysation enzymes, digestion enzymes that break down the proteins and leave behind the nucleic acids. So today, people don't typically have to do that step for separation unless they're doing special types of PCR. Typically now you can do it in a single step with an enzyme that just breaks down the cells and leaves behind the nucleic acids. And if you're looking at cell-free eDNA, which I mentioned that I like to look at, you don't even need the lysation. You can just run it through the filter and then straight into your machine. And that greatly simplifies and reduces the cost of the analysis. But when you're going to run PCR in the field, practicalities really drive the purpose, and you have to think about costs. I loved, the idea to run hundreds of detections. It's wonderful concept, but if you're paying $20, $30 a sample, you can bankrupt the U.S. Fish and Wildlife Service pretty quickly doing that. So, or state agencies. So how do you do this cost effectively? And so these are the extraction enzymes that we were looking at. This work was funded again by U.S. Fish and Wildlife Service. What can you do in the field that's cost effective? You can see up here that the cost per sample really relates to how much eDNA you're getting out of the sample. You pay more, you get more. But some of these aren't amenable to field deployments. They have centrifuges involved, and they're not something you're going to take into the field. This one under the McNeil column, is one we did trying to get down below $1 per extraction and requiring no centrifuges, but it didn't have a very good extraction efficiency. This one here on the left, Zygem, is the one that the community uses the most now. Still $3 a sample, and if you're measuring hundreds of cars a day, you're going to have problems paying for all of that. So I like looking at the free eDNA because you don't even need to pay for the extraction step. Questions people often ask is how sensitive or accurate this form of detection is. Well, it's just PCR, and it stands on the shoulders of giants for who identified the primers and probes, how we know they're specific. I think most of the community is using the Gingera primers and probes, and that's what I use. And yeah. So here's another recent deployment in 2019. That's when we got that Lake Mead data. We also did look at this data. We got micro gradients as we moved away from the shore. Position 1, there is mussel infestation observed. You see high concentrations of eDNA. And as we move away, you saw the concentration drop and then go back up again as we approached a buoy that also had zebra mussels growing on it. So we can see micro gradients with this instrument. This is the instrument that's deployed now at Inksdam National Fish Hatchery. They keep it close by where where their operations occur, and the instrument, you walk up to it with the sample, and then with this computer, you press the big green button and you run it through the machine. It's been particularly exciting deploying the machine and having it run by the people who did not build it. This is the first time we've ever done that in 10 years. And so it's a real learning curve about ways to break it and what can go wrong. This is a picture of the data analysis. I'm actually going to do something risky in this talk and actually show you that data analysis live. On this slide, you can see this is the data coming through from the machine, and you can select the region that you're interested in looking at, and then you can see all these zeros, all those nanoliter droplets that didn't have DNA, but then there's these 2 spikes that did have DNA, and then you can drag your threshold to differentiate between those 2, and it automatically tells you there are 2.27 copies per microliter of the reaction volume. This is lake water from Inks Hatchery above that lake that I showed you above the dam. And so we're seeing DNA from zebra mussel in that water. And yeah, this is the analytical tool that's given to the fisheries operators, and they can load their data and analyze it live and see the concentration of mussel at any moment. I'd like to go on to questions pretty quickly, so I'm not going to present much more about the machine, but this is where we're going to go from here. Like I said, it's been licensed. This is a image, as you can see, very commercial image of what it's probably going to look like, with the tube running into the water, sucking out of it the water constantly or every few minutes, and reporting the eDNA concentrations to the cloud. And we are incorporating internal RNA quantification with RNA heaters, so we'll be able to do viruses fairly soon. And we're just trying to get more deployments and with people who are uninitiated and untrained, and try to drive up that reaction speed. But me personally, as a scientist, I'm fascinated by the fundamental eDNA research you can do. Being able to go from gene copy numbers to organism abundance, saying there are 2 copies per microliter, how useful is that? And that step from gene copy number to organism abundance is sort of a holy grail of people who study eDNA, and having an on-site tool like this that looks at free eDNA can really help in that. So there's a lot of really unique science that can be done with this tool as well. Not just a useful tool in aiding management, but there's a lot of unique scientific questions. So with that, I would open it up to questions.
Great, thank you, Cody, for sharing your work with us today. So if you have any questions for Cody, please go ahead and put those in the question box or the chat. Let's see, I see one here first in, I have a couple for you here in the chat. I think Dennis and I are going to tag team these questions here. Okay, so first from Nate Owens asks, Cody, you mentioned testing residual eDNA before and after boat decontamination. Did that decontamination include the flushing of ballast tanks or engines? More specifically, was the water discharge from the ballast tanks or engine before and after decontamination tested for eDNA? Do you need me to repeat
so
any?
at Lake Mead, they have these, what I think are more expensive than your machine costs, that's for sure. But these cleaning stations that hot water spray nozzles. In this particular boat, they are flushing it with hot water, the engine compartment, the whole body of the boat, and I believe they're also doing bilge flushes as well with hot water. This water actually was just water that was dripping off of the boat. So coming out of there's a tube coming out of the bilge when they pull that plug, and that's water that came out of there. So.
Okay, let's see, I have another question in the chat and then we'll go to you, Dennis. So Tom Wolf asks, how do you interpret that last data set that was shown? Do those 2 spikes indicate strong evidence of dissonance?
Yeah, they don't. I don't think they do. I think that eDNA, molecular evidence in general, it's much easier to be critical of its false positives and compared to microscopy. I really believe that. So me personally, you know, if you believe the Mikey guidelines, they say you need at least 10 spikes per sample to be confident in quantification. But quantification is different than presence and absence detection. And, you know, we're able to run no template controls that come up blank. So when you see 2 spikes like that, there's something in that water. And whether or not that's something that needs to be taken is a whole other story. I kind of leave that, you know, to other people. Me personally, I focus on the instrument and the instrumentation, and we're seeing 2 positive detects. What that means is really context specific. You know, if it was a laboratory sample, they would be positives. But if it's an environmental sample, you know, what was the light level out? How positive is it? You know, what was the background microbial content? These are things that can degrade the eDNA. You know, these are all context specific. So I don't like to say we should take action based on something as low as 2 copies per sample.
Okay, thanks, Cody. And, you know, Adam on Thursday is going to be talking about structured decision-making eDNA, so we can have more chats about that later in the week. Dennis, do we have any more questions in the question box?
Yes, we do. From Linda Shaw. How is the free eDNA information being used to prevent infestation of fish hatchery water containing the catfish being transported on?
Yeah, that's a good question. They have a procedure there, a cleaning procedure, before those fish are allowed to go out. And so they're using this instrument to look at that water in the tank as a function of time as they process it. And yeah, that's how they're using it. And so they're interested if they're getting any positives at all at the end of that. You know, it would cause them to go back and possibly not ship and go back and look at it and take longer to clean them.
And then from Tim Cunningham, great talk. In your opinion and experience, is Dracaena DNA evenly distributed in a water body?
Yeah, no. So I'll put it out there for us so that we can keep this in our minds throughout this talk. I think that there are, you know, the patchiness of eDNA is phenomenal. And I think the number one things that drives its spatial distribution is the organism behavior. And in this case, you know, are they you know, spawning? Are they you know, what are they living like? At what stage are they in their development? So the number one thing is the organism behavior. And then after that is flow. eDNA, even free eDNA, is not a floppy molecule. I mean, it's not a small molecule. It's floppy. It doesn't diffuse by Brownian motion. It is driven entirely, almost entirely by the turbulence and the flow of the environments it's in. So, you know, I heard Adam Sepulveda ask the question of Megan earlier. You know, he's one to really speak to that patchiness, but it can be, you know, you can be 6 inches away from a source and not see it if the flow is such that it's taking the fresh water into your detection event. So it's very patchy, driven primarily by organism behavior, secondarily by flow. Then after that, I think what drives its distribution is the background microbial content. We have evidence now that says that if the water is very clear, that it's nutrient limited, then the background microbes are starved for phosphorus and they consume the DNA very rapidly as a food source. So, and then after that, I would say it's the light levels. I'd say how much UV light it's exposed to can degrade it and break it down rapidly. So it's a very patchy molecule. And so when you have a machine like this one that's detecting at a single point, if it's fixed, although it can be used and be mobile and you can explore that patchiness and do heat maps and gradient mapping. But when you sample at a fixed location, you're not doing a TONET or something like that. This machine can overcome that by sampling every few minutes, 24 hours a day, 7 days a week. It can sort of overcome partially that issue through its high frequency sampling. But no, it's eDNA patchiness is a great subject to think about.
And then we have 2 more questions. Do we have time, Leah?
Yes, we do. Go for it.
Okay, from Tom Booz, how will it be secured? I'm assuming it's some of the.
Well, secured, yeah, as a guy who builds instruments, I tell you, the thing you can never overcome is the human factor. You know, if somebody, I had to build a machine once that was shotgun proof, you know, this thing will not be, it's impossible. If somebody wanted to come and steal this machine, you're either going to have a cage around it or inside of something where people can't come up to it. But there's the other aspect that I think the question was probably more addressing, which is that it can be on shore with a tube running into the water. It can be fixed on the back of a buoy. It can operate at a variety of angles. Initially, we were doing impact studies and G-force constraints, and I've kind of shied away from that, you know, focused mostly on making it work stably, even when it's held fixed on a table is hard enough. Getting it to work in a really a high impact environment is the next level that now that it's being commercially developed, that is something that they're really focused on. But it can be mobile.
And then from James Dominguez, Cody, can you talk about determining if the DNA detected has been alive recently, in quotes, or
James,
if introduced from a boat that wasn't alive?
I owe James an email, man. He's so thank you. Glad you're here listening. I'm glad you asked the question, James. Right. So one of the neat things is a lot of people talk about how, you know, measuring DNA doesn't tell you whether it was alive or dead, because the genome doesn't do that, and we'd have to get to RNA, because then at least you know it was expressed fairly recently, and that you would have a much better chance of knowing whether it was an active organism with RNA than you would DNA. And I think that's fairly usually true. But one of the neat things about studying free eDNA is at least we have a chance, because there it degrades so much more rapidly, it's consumed so much more rapidly. The only reason you're going to find it is if a cell recently lysed. And so that means there was at least an intact cell there within the past, you know, hour or so. So it gets you a lot closer to viability using DNA without having to go to RNA for viability detection. But that's another interesting rabbit hole right there.
Then we have one more from Jeff Adams. How easily could it be used for detection of other species?
Yeah, this machine is totally, we specifically, I should have talked more about this point, which is that we didn't build this machine for a human health market. You know, I know I was introduced saying, you know, taking biomedical tools and bringing them to eDNA detection, but the fact is I didn't really ever care about the biomedical detection. And this machine would not get FDA approval because it has no hardware consumables. So there is a non-zero mathematical chance of cross-contamination. I mean, we can run controls to make sure we're not contaminated, but the FDA sort of requires there be consumables. So we specifically have no hardware consumables. This was developed for environmental detection. And also in that vein, we specifically have no proprietary reagents. This is just PCR. This is just, I can run CyberGreen, I can run TACMan probes. So if you have a primer probe set, this machine can be preloaded with that set, the commercial machine, which operates as a smoke alarm, or you can just walk up to it with whatever primer probe you want. And so it's very easy. Every sample, the new machine is a 3-color multiplex. We can do 4-color multiplex, and you can also walk up to it with a different gene target every single sample.
And then we did have a question from Philip Matson, but Philip, your question didn't type out all the way. So I don't know if you want to try to get back in there and ask that question. Otherwise, that's all we have, Leah. I don't know if you want to go back to some questions from previous presentations that we didn't get a chance to answer.
Okay, stop sharing.
Sure. Yeah, if we want to, yeah, thank you so much, Cody. That was great. Your presentation, very interesting. And I guess if Phil's question comes in, we can loop back to it. So yeah, sure, Dennis, let's go. I know there was a couple that we didn't get to from Nate's, the first presentation, to touch on. I think Chris Moffat had one maybe, and
yeah, I think Chris, it looks like maybe she's left, but she asked, did you include assessments of efficacy in trials with known infestation?
Yeah, that's a great question. And this is part of the difficulty of evaluating what how effective the normal decontamination protocols are. The one thing we know is based on research, quagga and zebra mussels in all life stages die at a certain temperature with a certain exposure time. So we know we are far exceeding those. So theoretically, we should be 100% effective because every part of the boat that actually gets wet while it's in the reservoir also gets wet in this hot water decontamination dip tank. We are starting to do a study right now looking at the veliger density in the tank water after one decontamination, after two decontaminations, and look at that over time, as well as the filtration system kicks in on a lot of those veligers that they're discharged during the decontamination process or removed from the tank water. So we had hoped to have some of that information available to present today. We didn't get to it. It will be coming. We'll probably do a smaller study as we shut down the tank this season, and then probably a more comprehensive study looking at that come the spring when we start opening that up for public use, probably sometime late April, early May. Good question, though.
And then looks like, Aaron Martin, have you been able to analyze the effectiveness of the dip tank? Which I think we've kind of alluded to already a little bit, but.
Yeah, same thing. We're mostly concerned about, and others have expressed this concern, whether we are essentially pumping boats full of veligers at a concentration greater than what they would be exposed to in Lake Powell. We don't think that's the case. In any event, we know those veligers are dead just because they're sitting in hot water. But we also want to look at that more critically and see if that changes over time, see how effective the filtration system is, whether we need to maybe revamp that and look at something that filters more out more frequently. All great questions, all things we're hoping to address. The primary focus this first year was just making sure that all the systems were operational and that we had the decontamination protocol down as to how this would work when boats are actually coming down into the tank and then also exiting the tank.
And then the last question I see from Quagga D, are other states accepting the dip tank decontamination?
Quagga D, that's a question for those states, I guess. As far as I know, every state that currently accepts a normal decontamination is also accepting a dip tank decontamination. I would hope that would be the case in the sense that we know this is far more effective than anything we're doing out there because we are completely filling all these systems with hot water. If anyone does have any concerns of this being not as effective as what we've done in the past, I certainly want to hear those and see if we can address those, because in our minds, we are far more comprehensive in this decontamination process than we have been in the past.
And maybe a follow-up to that, and I apologize if you've already stated this, but do you provide any documentation that demonstrates whether it was a dip tank decontamination versus a more traditional unit that we use with the fake lakes and earmuffs and things like that?
We currently do not, although we've talked about trying to figure out a way to put that into the regional database so that when other users of the database retrieve that record, there is some sort of tag associated with that decontamination record. I think that's probably something we're going to look at over the course of the off season.
Great. All right. Well, I think that's it for Nate. So thank you, Nate. And then Philip Matson was able to post his question for Cody. Is there a higher chance for false positives from free eDNA versus biologically bound DNA?
I'm thinking. I'm thinking.
Get the Jeopardy tune going on.
Yeah, I really don't know. We are just now studying the relative abundance of the two, even let alone, you know, their rates of false positives. So it's, I do know that free DNA is this is some apocryphal evidence, but when we sampled through a standard filter 3 liters of water and then did extraction, laboratory extraction, purification, and quantification, all of those steps are so inefficient that we were getting similar gene copy numbers as to just a few microliters of sample analyzed for free DNA without those processing steps. So you know, that would play into it. Their relative efficiencies of extraction are different, and you know, they're lossy. All of those steps in processing the water are lossy, and you get different loss rates for free DNA as opposed to cell associated. So it's actually a pretty complicated question, and I don't know the answer. It's something we could figure out together, Phil.
I think that's all we have, Leah.
Okay, well, awesome. I guess it's time for you to close it out. Let me bring up the, you can bring up the agenda, but go ahead and close this out.
Yeah, thank you to the presenters. Fascinating presentations and topics. Really appreciate your willingness to participate, and thanks for the great questions, very intriguing and thoughtful. And if today was any indication of what's to come this week, we're in for a great meeting with some of these presentations that are, like I said, are really fascinating. So we will sign off, and please, if any other questions pop up while you're having a cocktail this evening, feel free to email Leah or I, and we'll make sure that those get out to the presenters. And thanks to Tom Wolf for moderating the session. And we will see you all tomorrow, 12 Pacific, 1 p.m. Mountain, right? I think that's right.
Yes, you got it.
And Tom will kick us off in the morning, and we'll have Christiani moderating some emerging coastal issues and advancements. So thank you all, and look forward to the rest of a great week. Have a good night.
Thank you.